Okay, so for the specific parts of our renal corpuscle, we can see that there are actually other cells which are surrounding your tuft of capillaries, no? So here you would see your cells which are called your podocytes intimately attached to the endothelial, sorry, attached to the basement membrane of your capillaries, okay? Actually, these podocytes make up the visceral layer of your glomerular capsule. Okay? And then in between these individual capillaries, you would actually have your glomerular mesangial cells, which in some references, they would have different names, whether it's intraglomerular or outside the glomerulus. No? And then in the distal convoluted tubule here that is seen closely opposed to your afferent arteriole as well as your efferent arteriole. There is also a special group of cells in its wall, the wall of your distal convoluted tubule nearest your afferent arteriole, which are called your macula densa cells, no? So these macula densa cells, they will play a role in the control of the absorption of nutrients, as well as they are also producing specific hormones, no? And then aside from your macula densa cells, you would also have your juxtaglomerular cells, which are actually just specialized smooth muscle cells attached to the outside part of your afferent arteriole. No? Histologically, usually it is your macula densa cells which can be easily seen or observed under the microscope, no, because they look really dense, no, densely staining nuclei. So let's see later if we can identify them in actual histologic section. So here, you can see the histological version of this schematic diagram, and what we can observe is that it's very difficult to differentiate which one is a capillary and which one is a podocyte. But here you can see that the macula densa cells really look different from the normal cells of the distal convoluted tubule. But anyway, the combination of these macula densa cells as well as here the extraglomerular mesangial cells also has another name, no, depending on the reference. But in general, the combination of these all of these cells is called as your juxtaglomerular complex. Okay, so it's here. Okay, and here we can see again the glomerular capsule parietal layer, and then we can assume that the visceral layer is here, and then this is the urinary space. So again, these macula densa, and if the afferent arteriole is present, the special cells that are part of the afferent arteriole, together they form the juxtaglomerular complex, which is a complex that plays a role in regulating again the expulsion of filtrates and waste products, as well as the reabsorption of water, which can in turn have a function in blood pressure regulation. Okay, so again, this is a more specific schematic diagram of the different components of the juxtaglomerular apparatus, as well as the renal corpuscle itself. Okay, again, another photo, no? So what we can say here is actually in most of the sections, it would be the macula densa that is easier to spot, no? Um, not all sections of your renal corpuscle can actually see a very nice view of your juxtaglomerular apparatus. But when you view the actual slides in the lab, try your best to look for them. Here is another photo, actually from a research paper, which actually explores different stains, no? For the kidney, another type of stain aside from our typical H&E that is usually used would be your PAS staining or periodic acid shift staining, which employs a purplish stain that prefers the carbohydrate components, no? It usually stains the basement membrane of your lining epithelia. So, for the past staining, it's better because you can actually see the capillaries, no, where each capillary ends or starts. Okay. Silver stain can also stain the connective tissue. So again, the folded tuft of capillaries is more defined in these special stains, no? Of course, we can identify the capillaries from the glomerular mesangial cells as well as podocyte because within your capillaries, we can actually see, as with all other blood vessels, you will see the presence of your RBCs. Okay? Next, let's talk about the convoluted tubules, both the proximal and the distal. No, let's describe between the two. There's a great difference between their shape and staining color. Here you will see that in the proximal convoluted tubule, because it has a more absorptive reabsorption role, you would see that the apical portion would actually have this microvilli-like structure. No, what else? The proximal convoluted cells actually has like a more darker appearance because the basal part of each cell would actually have infoldings as well. Because again, these foldings would maximize the passage of wastes and nutrients from the inside of the lumen into the surrounding tissue or blood vessel. Okay, so the proximal convoluted tubules is more darkly staining and would have this fuzzy appearance in the lumen as opposed to your distal convoluted tubule, which is technically more lighter and simpler and has a more clear lumen than your proximal convoluted tubule. Okay? For so again, distal convoluted tubule, and if it has a complicated, more fuzzy luminal area, that would be your proximal convoluted tubule. For the pars radiata or this part, you'd have your proximal straight tubule, and then you would also have your um, thin descending limb, thin ascending limb, and thick ascending limb. And since the proximal straight tubule, so again, this photo is actually a cross section. Looks like this, no? And you would know that it is a straight tubule because it is contained within the pars radiata area. Okay? So you can see that the proximal straight tubule walls is almost the same as with your earlier, our convoluted, no? The proximal convoluted and straight tubule would have the same fuzzy and more microvilli formation in its luminal area. Okay, as opposed with your thick or thin, so more flatter cells, more cuboidal cells. The apical surfaces of the cells are not as fuzzy because they do not contain much of that extensive microvilli. Okay, again, for the thick, actually it's difficult to differentiate the actually it's possible to differentiate the proximal from the distal straight because of this pattern, no? Proximal would have this fuzzy part, and and the thicker one. And then for the thin limbs, so this thick ascending limb of the distal part would not have that fuzzy appearance. While for the thin limbs, this part and this part, it would have a more flatter set of lining epithelial cells. Okay. Then for the collecting ducts, this one, you would have a more taller cells, and then the collecting ducts would have the more cuboidal and plumper cells. Okay. Here in this photo, you can see the portions of the thin limbs with the thinner walls, and here the collecting ducts. Okay. And you would be sure that it's the collecting duct if it has a bigger lumen than the straight parts of your loop of Henle. Okay. Again, it's almost very tall cuboidal lining epithelium. Okay. So this is a summary of the different changes in the lining epithelium of each segment of the uriniferous tubule, as well as the actual function and substances that they are reabsorbing, and as well as the um, substances that they are secreting. So you will see that, for example, in the proximal convoluted tubule, it needs a lot of those microvilli because it really reabsorbs a lot of things. While for the distal convoluted tubule, it, aside from reabsorption, its main function is for the secretion. Okay? So that's why it doesn't have an extensive microvilli system. And then for the other parts, you would have just additional reabsorption, no, of some items. And then some areas would be again for both reabsorption of water and some electrolytes and some ions. And then finally, some areas would just be a conduit so that the filtrate or the urine can finally exit in outside of the body via the ureter into the urinary bladder and finally into the urethra. Okay. So and then in this last part where the collecting ducts will actually empty out into the ureter, that is your renal pelvis area, this space. Okay. And here you can see that this area which is connected to your ureter is already transitional, has already a transitional type of lining epithelium. Another term for it is urothelium because the lining epithelium of the urinary system is almost always your transitional epithelium. Okay, it's like in the respiratory system, the respiratory epithelium is actually your pseudostratified lining epithelium. Okay, so again, these are your collecting ducts, and they will eventually empty into the renal pelvis and finally into the ureter. Okay? What else, no? So we talked about the uriniferous tubule, but as I said earlier, let us not forget the blood vessel component of your kidney. So these are your arteries and vein, no? Depending on where you see the specific blood vessels, they will change their name. So the usual blood vessels that you can see, if it's between the cortex and the medulla, and it's oriented in this way, or parallel to the capsule, then usually that would be your arcuate arteries, no? And then the arteries that are in between the lobes, you call that your interlobar blood vessels as well. And by now, you should be expert in differentiating which blood vessel is a vein and which one is an artery. Okay, so here you can see that this is the cortical area and this is the medullary area. And this part actually corresponds to this part. Okay. Okay, now let's go to the tubular part of the urinary system. So for the ureter, again, It has a transitional lining epithelium, and then you would have your lamina propria that is continuous with the submucosa, and then you would have your tunica muscularis, which would have a inner longitudinal and outer circular layer, and finally you would have your tunica adventitia or serosa. So another image showing the different layers of your ureter. Some references would actually just call this lamina propria, and that's okay as well. But I think it's better to use the lamina propria dash submucosa term. And here we can see that the transitional epithelium is basically a stratified version with the top cells changing shapes, no? Here it looks like it's balloon shaped or polyhedral or cuboidal. So it means most likely this part of the ureter is not filled up. So it's empty. Okay? For the urinary bladder, it's almost the same banana, but you would see that the muscularis layer is more developed. For the epithelium, it would still be transitional epithelium. Okay? So again, if the top cells of your transitional epithelium is cuboidal, circular, or polyhedral, or balloon shaped, it gives us an idea that the tissue that was collected is in a empty bladder, while if it is stretched and the top part is looking like squamous flattened cells, then most likely the organ was full when it was collected and fixed and processed for tissue sectioning. Okay? For the urethra, it's also your transitional epithelium. Okay? And depending on where the urethra is in the part of the reproductive system, in this case, the male reproductive system, its lining epithelium can change. Okay? The more that is nearer to your urinary bladder is still transitional, then eventually it will turn into columnar, and then finally, if it's nearer the outer edge, it would have your squamous epithelium. Again, the favorite stratus squamous epithelium, the more sturdy epithelia that is lining parts of organs or parts of the body that will be more in contact with the outside and environment. Okay, so here you can see in different areas. Now, for example, in this area, still the lining epithelium would be transitional. Okay, for the avian kidney, there's basically just a difference in the arrangement of the medullary parts and the cortical parts. No, the medullary parts come in discrete islands, no, and this called your renal pyramids as well. Okay, and areas that would look like areas of your collecting ducts or tubules are also present. And another key point is that there is no renal pelvis. So it's immediately the ureter that is immediately in contact with your kidney. Okay? No renal pelvis. And then what else? Another distinguishing characteristic of your avian kidney is that you would have different types of nephrons or yes, or of nephrons, no, you would have renal corpuscles that would have smaller sizes as well as bigger ones, no. Um, and then the arrangement of the blood vessels is also quite different, no. Here you would see that there's the sorry, these are the I think this reference is wrong. This is actually part of the uh renal vein which empties the cortical areas, no. Anyway, the one that is nearest to this structure, you would have your smaller renal corpuscles. These smaller renal corpuscles, they don't have the loop of Henle, so they are more simpler and more primitive, thus they are also called as your um reptilian nephrons, while the bigger ones, they would be more structure wise, they look like your mammalian nephrons. So these mammalian nephrons, they would have your more extensive loop of Henle. So again, when you view a histological slide of the avian kidney, aside from their general difference in the arrangement of the medullary and cortical tissue, you would see that there are different sizes or different sized populations of nephrons. Okay? So again, you would have your smaller corpuscles versus your bigger corpuscles. The bigger corpuscles would correspond to the mammalian counterpart, while the simpler, more primitive corpuscles will correspond to your reptilian nephrons. Okay, and that's it. And attendance.